Hello everyone and welcome back to Biotic World channel. I am Sheetal and in today's video we are going to explore one of the most exciting field in plant biotechnology, micropropagation. Whether you are a beginner just diving into plant tissue culture or looking to expand your knowledge, this video will guide you through the process from basic concepts to advanced techniques. Let's get started. So, what exactly is micropropagation? In simple terms, it is a method used to grow plants from small pieces of a plant material in a sterile, nutrient-rich environment. This technique is incredibly important in plant biotechnology and agriculture because it allows for the rapid multiplication of plants, a process that's faster, more efficient, and more reliable than traditional methods like cutting and grafting. An explant is any small piece of the plant used to start the process. It could be a leaf, stem, root, or even a single cell. These explants are cultured in a specially prepared medium that contains all the nutrients they need to grow. Let's break down the entire micropropagation process into several key steps. These are the foundational processes that apply across most types of plants. First, you need to choose a healthy donor plant. The plant should be disease-free and free from pests to ensure the success of your tissue culture. The type of explant you choose will depend on the species of the plant and the goal of your micropropagation. Sterilization is a critical step to prevent contamination by bacteria, fungi, and other microorganisms. The explants are typically surface sterilized using chemical agents like sodium hypochlorite or alcohol. This goal is to kill any unwanted organisms without damaging the plant tissue. Once the explants are sterilized, they are placed on a solid or nutrient medium containing nutrients, sugars, vitamins, and plant growth regulators. These plant growth regulators, including auxins and cytokines, play a crucial role in determining how the plant will grow, whether it will form roots, shoots, or calyces. For example, auxins promote root formation while cytokines encourage shoot development. A careful balance between these hormones is essential for successful tissue culture. Once the explant is placed in the right medium, they begin to divide and produce shoots. The goal here is to induce the production of multiple shoots from a single explant. This stage is known as the shoot multiplication, and it's where the plant mass grows significantly. After the shoots have developed, they need to form roots before they can be transferred to soil. To do this, we place them in a different medium that encourages root development. This is known as the rooting phase. Finally, after the plantlets have developed both shoots and roots, they are carefully removed from the sterile environment and moved to a controlled environment like a greenhouse. This process is known as acclimatization. The plantlets gradually adapt to the external conditions like light and humidity until they are ready to be transplanted into the field or garden. Now that we have covered the basics, let's explore some advanced techniques in micropropagation that have opened up new possibilities in plant biotechnology. Somatic embryogenesis is a process where somatic cells instead of reproductive cells are induced to form embryos. This is a more advanced form of micropropagation and is especially useful in producing large number of genetically identical plants from a single cell. Another major advancement in micropropagation is the introduction of genetic engineering by using techniques like agrobacterium-mediated transformation or gene gun technology. We can introduce new genes into plants. This allows us to create genetically modified plants with enhanced traits such as resistance to pests or diseases or improved nutritional content. Somaclonal variations refers to genetic variations that occur in plants grown from tissue culture. Sometimes these variations can lead to desirable nutrients such as disease resistance or increased yield, which can be useful in breeding programs. However, it can also result in unwanted changes, which is why careful monitoring is needed. In large-scale micropropagation, bioreactors are used to propagate plants in a controlled automated environment. These bioreactors can handle large volume of culture medium, and plants are grown in a submerged culture, allowing for quicker multiplication of plantlets. This is especially useful for producing millions of identical plants in a short period. For long-term storage of valuable plant material, cryopreservation is a vital tool. Plant tissues or embryos can be preserved at ultra low temperatures, effectively stopping biological activities. This method is used for conserving germplasm, rare species, or valuable cultivars. Micropropagation is used in many areas of plant production. Some common applications include mass production of disease free plants. For crops like bananas, potatoes, and strawberries, micropropagation ensures that plants are free from diseases that might affect traditional propagation method. Second, conservation of endangered species. It's also used in conservation efforts to propagate rare or endangered plant species that might not be able to reproduce in the wild. Third, clonal propagation in horticulture. Many ornamental plants such as roses and orchids are propagated through tissue culture to maintain consistency in their appearance and quality. Despite its many advantages, there are challenges in micropropagation. The initial setup costs can be high, and maintaining sterile conditions requires constant vigilance. Additionally, some plant species are more difficult to propagate than others due to their unique biological characteristics. However, with advances in automation, bioreactors, and genetic modifications, the future of micropropagation looks incredibly promising. New techniques are being developed to improve efficiency, reduce cost, and enhance the diversity of plants that can be propagated. That wrap up our deep dive into micropropagation from the basics all the way to advanced applications. I hope this video helped you understand the incredible potential of this technology in the world of plant biotechnology.